Hello, everyone, and welcome to today's live broadcast, Live Cell Analysis, Affordable and Easy, presented by Christian Oberdanner, PhD, and Cedric Muller, PhD. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Tecan. Tecan is a leading global provider of laboratory instruments and solutions in biopharmaceuticals, forensics, and clinical diagnostics. The company specializes in the development, production, and distribution of automated workflow solutions for laboratories in the life sciences sector. Its clients include pharmaceutical and biotechnology companies, university research departments, forensic and diagnostic labs. As an original equipment manufacturer, OEM, Tecan is also a leader in developing and manufacturing OEM instruments and components that are then distributed by partner companies. To learn more about Tecan, please visit www.tecan.com. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know you're having a problem. Thank you. Now I'd like to introduce Dr. Oberdanner and Dr. Muller. Dr. Oberdanner is a product manager at Tecan, Austria. He studied molecular biology at the University of Salzburg with a strong focus on cell and tumor biology. Dr. Oberdanner began working for Tecan in 2005 as an external scientific consultant and joined the company permanently in 2006 as an application specialist. As a product manager, he focuses on the company's multimode microplate reader portfolio. Dr. Muller and his SMALTIS co-founder Sophie Gaynard are young life sciences and health PhD graduates from the Tecan Bacteriology Laboratory directed by Professor Patrick Plesiot, where they studied the antibiotic resistance mechanisms of Pseudomonas aeruginosa. Since 2014, their company has provided its expertise in microbiology and molecular biology to help its clients accelerate their research projects. Please join me in welcoming Dr. Oberdanner and Dr. Muller. I will now turn the presentation over to them.
So, thank you for that nice introduction, and a warm welcome and hello to the audience also from my side. What can you expect from that educational webinar today? Very briefly spoken, it will be divided in two parts. In the first part, I will introduce Tecan's solution to easily establish live cell analysis in a lab without blowing your budget. And then I will also give you a review of what we have done on live cell analysis from an application perspective. This will include how to automate long-term cell viability, cytotoxicity and apoptosis assessments. I will talk about how to automatically monitor cell migration and how to perform monoclonality verification with the system's integrated imaging module. I will explain how to normalize signals from cells to the cell mass per well using Confluent assessment. And I will talk about the importance of whole well analysis in fluorescence bottom readout. Then in the second part, my dear co-speaker Cedric will talk about his experience with our solution and share all the exciting methods he established on live cell analysis with bacteria and eukaryotic cells. So biology is certainly always about investigating processes of life. So everybody of you will know, and I'm sure has already experienced, that assaying cells in an appropriate window of time, for example, to investigate compound-mediated cytotoxic effects, that can be extremely challenging, because you first of all need to keep your cells healthy during the whole investigational period. You need to choose the right assay and method that allows you to perform real-time and live cell analysis, and those methods should certainly produce high quality and reproducible results. Then you need to acquire all that data constantly over time, and you should not lose any data in between. So the very well-known overnight gaps in a kinetic measurement can be really critical, and you can lose important information. Unfortunately, the time courses are relatively long, so more than 24 hours is standard. Up to several days is necessary to investigate biological processes. And as always, the whole workflow, the whole procedure needs to be relatively cheap and should have also a minimum in manual inputs, of course. So basically two solutions or two approaches are well known. The one is what I did during my PhD study, sleeping in the lab, getting up every hour and acquiring data points to get a full data set over several days. Or you simply do it with a robotic system, but then budget should not be any issue in your lab. This now totally changed with Tecan's solution for cost-efficient live cell analysis, which is the Spark multimode reader. The Spark multimode reader is a system that is fully modular, upgradable, and it includes all important high-end detection, measurement modes, fluorescence, absorbance, luminescence, alpha screen, and so on. But more excitingly, it includes also features that support live cell analysis. And this is what I will explain to you in more detail in the next couple of slides. So the first element that we incorporated in the system is environmental control. We need to keep the cells healthy during the analytical experimental time period. And the first element is the temperature control. For temperature control, we made sure to isolate the plate transport chamber from the optics in the system to guarantee a best possible temperature distribution in the system. That allows us to specify a cross-plate uniformity of plus minus 0.5 degrees Celsius. We use a top heating foil to avoid condensation on plate lids, and most excitingly, we have the so-called Takehome module, which is the first air condition for a multimode reader, and this enables us to keep a stable temperature between 80 and 42 degrees over time. And this, of course, has a lot of important and interesting effects, but one example is that you can perfectly handle now non-mammalian cells which have a temperature optimum far below 37 degrees. For instance, insect cells belong to this group. The second important element when it comes up to environmental control implemented in the reader is gas control. So we can control the CO2 and O2 level inside the plate transport chamber very accurately and in a very broad range. It's all fully integrated in the system, and everything is fully controllable via the Spark control software, which is the reader control software of the system. We can even log CO2 and O2 data over time so that you always have the full control what is going on inside the system. Notably, O2 can be regulated down to 0.1%, and we can keep the hypoxic conditions then between 0.1 and 0.6% over time. And this enables us to really provide a system to do physiologic relevant studies even under highly hypoxic conditions. The third element, and that is something that is a patent pending technology from Tecan, is the humidity cassette. So we invented a cassette, a metal cassette, that incorporates a mode that can be filled with deionized water. And in this cassette, you can put then every possible microplate in SLS format. And by closing that cassette, the atmosphere inside the cassette will be passively humidified by the evaporation of the deionized water, and this will then avoid the evaporation of the liquid in the sample well. Certainly, this does only function together with Tecan's unit lid lifting system, because for measurement and for other actions you need to remove the lid of the humidity cassette. And for this we invented an integrated lid lifting system, so the Spark can de- and relid microplates inside the reader, not only the humidity cassette, but also every other possible microplate lid. You simply need to stick a magnetic pad on it, and the lid lifter will be able to handle also the plastic plate. This exciting feature is accomplished with some other features that we all put in the group and call smart automation. That is first of all a dispenser module that is equipped with a built-in reagent heater and stirrer, but also software features, the kinetic conditioning feature that allows us to perform actions as soon as a certain value is reached inside a control well, a very exciting feature for kinetic measurements, and the open kinetic feature, which allows users to pause and resume kinetic experiments, which simplifies multi-user operations. So these features, the lid lifting, the dispenser module, the software features allow us to smartly automate the workflow, and that does not mean that the workflow or that we can increase the throughput with this, but we minimize the manual inputs and make it much more convenient to do long-term kinetic measurements, especially with cells. Another element that is important for live cell analysis is the integrated cell imaging module in the Spark reader. It's a 4x bright field-based imaging module, cost-efficient but very easy to use solution. You can use it for cell counting and viability checks of suspension cells, trypsinized cells in slides, or you can measure the confluence level and take images in bright field from adherent cells in a microplate well. We also offer a live viewer, which is a kind of microscope mode integrated in our software. Finally, when we talk about cell-based assays and long-term in vitro studies performed inside the reader, we need to talk about fluorescence bottom readouts, which is the most important measurement mode for cell-based assays in general. And here we provide several elements that help to push the performance of your assay. This is first of all our recently launched Tecan microplate dedicated for fluorescence bottom measurements in 96 and 384 well format, which was optimized and tested together with the Spark multimode reader. But also software features help us to improve the quality of your data for fluorescence bottom readouts. This is the very interesting function called optimal read that allows us the whole well analysis of the microplate well, and I will show you some exciting data as well at the very end of my presentation. But basically this feature allows to record the signal from each and every cell inside a well, no matter what the plate format is. So even an 84 well or a 24 well plate can be analyzed, fully analyzed with our system. We also offer set optimization for fluorescence bottom readouts. That means we can optimize the set focus for fluorescence readings, which makes the system very sensitive for cell-based assays. And we have a feature that we call high-definition well area scanning. This is a feature that allows us to create a low-resolution fluorescence image of the entire microplate well with a 100 per 100 measurement pattern. But here, we don't use a camera system. We use our normal fluorescence bottom readout. So honestly, this takes some time and is, of course, just a low-resolution image of your cells inside the well, but still can give you an idea what is happening. So enough with the technical details on the reader itself. Let's talk about what you can do with the system. And here I want to start with a collaboration that we did together with Promega. And here in that study we describe a method for automated kinetic monitoring of cell viability, cytotoxicity, and apoptosis. What we used are the kits from Promega designed for real-time analysis, which is the real-time glow assay to detect viability, the cytotox screen assay to detect cytotoxicity, and the Cas-Pass glow assay to detect apoptosis. We cultivated K562 cells for over 72 hours inside the reader, of course using all the features I just explained, humidity cassette, temperature control, CO2 control, and investigated viability, cytotoxicity, and apoptosis over time. For the real-time glow and the cytotoxicity green assay, you just add the reagents at the very beginning, and we left the lid of the plate on the plate for over 72 hours and just did periodically the respective measurements with our reader, so luminescence and fluorescence measurements. For Cas-Pass 3.7 measurements, we needed to inject the Cas-Pass 3 reagent, and this we did automatically with our injectors. We pre-warmed the reagent and then at certain intervals injected the Cas-Pass 3.7 reagent. We incubated 30 minutes inside the reader and then measured the luminescence. So also this assay was fully automated without any manual input needed. And certainly this was just possible because we have the lid lifter system that allows us to unlid the plate, inject something, and then relid it again. The researchers at Promega, they also did a second experiment where they tried to investigate the potential of the kinetic conditioning function. And here they used a cytotoxicity threshold for an automatic Cas-Pass 3.7 injection. And as you will find out when you study that paper in more detail, that also perfectly worked out. So at the end of the day, we concluded that using just the onset of cytotoxicity as a marker of apoptosis, it is more difficult to correctly identify the apoptosis induction window for a specific drug and or dosage, and that potentially leads to the incorrect mode of action being assigned. So it's always very important to study all that markers over time without loss of data in between. The next study I want to review on is very similar to the first one. It's again about measuring metabolic markers in cells over a long period in time. But in this case, we work together with researchers of the University of Heidelberg and a company called Promocell, which provides assays very similar to those of Promega. And we investigated the differentiation of human mesenchymal stem cells. We used ready-to-use differentiation media and monitored then over 60 hours differentiation to adipocytes, chondrocytes, neurocytes, and osteocytes. And during differentiation, we measured the cell viability constantly with a fluorometric cell viability kit based on resveratrol. But in addition, we also measured cytotoxicity via LDH and ATP levels using a bioluminescence cell viability kit from chromosomal. What we found is that the cell counting and plating and culturing of stem cells for several days with the Spark worked out fine. Yes, you understood it correctly, cell counting and plating, because those researchers of the University of Heidelberg, they used the counting to count the cells, but then they also wondered if they could inject them with our injector system, so if they could be plated for culturing. And that worked out also fine. So it was everything was very sterile, and the cells perfectly grew after injection inside the reader. We then monitored the differentiation over 60 hours and measured viability, and here we found that resveratrol was non-toxic to stem cells, but a very sensitive approach. And early after induction of the differentiation towards adipocytes, we found a significant elevation of metabolism and ATP content. In contrast, the osteogenic, the chondrogenic, and the neurogenic differentiation significantly reduces the metabolic activity of MSCs. The next two studies, or better 3 studies, all relate to the imaging function of the Spark reader. In a first study, we wanted to investigate the potential of the system to monitor cell migration. For this, we observed the migration potential of 2 human biliary tract cancer cells. One was reported to be to not migrate at all, and the other was reported to show migration properties, and we simply wanted to prove it. Again, we did this together with a collaboration partner at the private medical University of Salzburg. And we used the radial cell migration assay together with our system. This assay is a very nice thing because it creates a cell-free area in the middle of a fully confluent well, and you can perfectly then monitor this cell-free area for cell migration over time. To stop cell proliferation, we changed to FCS-free media and then observed the gap closure either manually with the microscope, so that means taking the plate out every hour, taking the image manually, and then doing the image analysis manually. And this we compared certainly with the automated approach in our reader, where we used again humidity cassette, the gas control, temperature for the whole experimental period, and imaged the center of the well with our imaging function and used the inverse confluence value to plot migration. And as you see, the results are perfectly comparable, the automated approach to the manual approach, but notably with the manual approach you lose a lot of data points in an overnight gap, whereas with the Spark you can constantly measure the repopulation of the cell-free area and don't lose any data points at all. In a second experiment that is explained in that note, we show that the confluence level perfectly correlates with the ATP level in the same well, and this leads me to the next topic, because you can use the confluence estimation or the confluence assessment in our system to normalize on cell mass. We describe this effect or this potential in a technical note that you can download on our homepage certainly, and we show that GFP and confluence signal perfectly correlates. Here we showed between 10 and 60%, but we also proved that between 10 and 90% with other assays like the BCA assay or like the ATP measurements. So this clearly shows that just by measuring the confluence level of the whole well using the Spark, that can be used to normalize on every possible assay signal coming from the same cells in the well. And what you skip is, of course, an additional assay. You save reagents and money, and that is very convenient and very helpful in an overall workflow. The last application that is on imaging is very quickly explained. We wanted to test the potential of the reader for semi-auto or for monoclonality verification. What we found basically is that yes, it works. You can automatically record the images as the system can make whole well images. You can then, after a couple of days, easily track back to find single cells in a well. So the image resolution, the image quality is good enough to also support these kind of applications. Certainly, it's something in between the manual approach and a dedicated system for clonality verifications like the CSI from MD. The last application note, or the last study our researchers here at Tecan did, I want to explain to you, is about fluorescence bottom readouts in general. And here we show the importance of the optimal read function. As you can see on the very top image, these are fibroblasts, and they do not grow homogeneously in an 84 well plate. They tend to grow from the middle. And certainly if you just measure that part in the middle, you get a wrong result that is misleading. And we showed in that application note that in 84, but also in 96 well plates, the CV of your signal dramatically drops as soon as you use optimal read instead of single read. And that, of course, directly correlates with a very high well-to-well uniformity and an excellent sensitivity for cell-based assays as soon as you make sure to analyze the whole well area and not just a small part in the middle. Good. So finalizing my first part, I want to give you some take-home messages. So first of all, modern multimode readers offer more than just absorbance, fluorescence, and luminescence. They enable for automated live cell analysis, and this is really a cheaper, let's say, and more cost-efficient approach than investing in high-end robotic systems or high-end live cell imaging systems. And of course, it's better than staying in the lab for the whole night. In addition to well-established functions such as heating, gas control, cell imaging, the Spark is equipped with some unique features, including humidity control, instrument cooling, lid handling. And this allows to simultaneously maximize data quality and minimize hands-on time. We could show that in several collaborative studies, we could show that monitoring biological processes is very important to get out the right results. So by doing this with the Spark, you can save time, money, and especially it's much more convenient for you. Easy to establish and cost-efficient procedures for imaging-based cell migration and monoclonality verification can be achieved using the Spark. I think that clearly came out. And an important message again at the end, bottom reading, it's standard for every multimode reader, but just special software features that allow you to analyze the whole well will give you reproducible results with a good well-to-well variability, and thereby offering a perfect and excellent sensitivity of the system. If you are now curious and you want to know more about the Spark and about other applications that our scientists and our collaborations partners did with the system, just visit our homepage. And with this, I'm really happy to hand over to my co-speaker, Cedric. Thank you.
Well, hello. So before starting, I would like to thank you, Christian, for introducing me and for giving me the opportunity to present our company named SMALTIS. So my speech will be divided in two parts. In the first part, I will introduce to you our company and its capabilities. And in the second part, I will present you some of our services where we analyze bacterial cells, live bacterial cells, and how the Tecan reader improve our capabilities. So, SMALTIS is a contract research organization created 3 years ago, and we are specialized in the fields of microbiology and molecular biology, and we provide customized services to our customers to increase their research capabilities. For this purpose, at SMALTIS, we deliver scientific expertise combined with innovating tools in order to accelerate your research programs. As you can see, SMALTIS is located in Besançon. It's located in the east of France, next to Switzerland, around 1 hour by car. And we are located near the Jean Mingeot Hospital and the Faculty of Medicine to have a very important work network and collaboration. So to provide high quality services and to guarantee our results, we have acquired on our own financial resources a fully equipped biosafety level 2 laboratory. And thanks to this investment, our company has a remarkable capacity to adapt and a total independence. Regarding the team, our staff has a successful academic background and many years of expertise in microbiology, molecular biology, cellular biology, and antibiotic resistance. So to introduce you our team, the team is made of 3 PhD holders, including the 2 founders, so Dr. Sophie Guénard and myself. The team is composed also of 1 engineer and 1 apprentice. And SMALTIS benefits from the advice of Professor Patrick Plezia, which is a very internationally known microbiologist. So, relying on our research abilities, we developed our activity according to an interconnected mixed economic model, which enabled us to set up a diversified and innovative toolbox. So, as you can see on this figure, SMALTIS team focuses on developing innovating biological tools in order to improve our service offer. And in this way, SMALTIS is able to collaborate with new customers who have most often new method requirements, and so it involves the development for us of new tools. Hence, at SMALTIS, we operate globally in the R&D project or in the research process by offering unique customized services in microbiology and molecular biology, specifically designed from the request of our customer. And I will try after to show you some of these services. But before to give you an overview of our capabilities, I'm going to introduce firstly our microbiological tools. So they are divided in 5 groups. The first one include all the methods to characterize microorganisms. So for instance, at SMALTIS, we are able to identify all bacteria, yeast and mold by using from culture method to next generation sequencing technologies. Then, when microorganisms are identified, we're able to characterize some of their properties, such as their growth rate or their antibiotic resistance phenotype or their capability to produce virulence factors. We can also discriminate several microorganisms by using genotyping methods such as pulse field gel electrophoresis or MLVA method. Secondly, we have developed specific tools to customize the genome of gram-negative bacteria. So we can propose to delete specifically a gene or to replace it by another one without leaving any additional DNA or resistant cassette. But I'll give you an example of what we do with this tool later in the presentation. Thirdly, we have developed specific tools and methods to study the skin microflora. And this method allowing us now to be the only company in Europe which is capable of studying the skin microbiome directly on healthy volunteers. Fourthly, several tools have been developed to evaluate how bacteria can adapt to a specific compound, either at the molecular level by measuring gene expression or at the phenotype level by measuring resistance level. Finally, eukaryotic cellular models were carried out to evaluate the bacterial adhesion, invasion, and cytotoxic properties, to analyze the cellular response to infections, and finally, to screen compounds for their anti-infective properties in eukaryotic infection models. Regarding our molecular biological tools now, they are divided into three groups. The first one pools method to prepare nucleic acids from different sources, such as blood cells, pellets, biopsy, and so on. The second one regroups method to quantify gene expression by using qPCR, RT-qPCR, and also a new PCR method named LabPCR. We are also able to do blotting experiment or in vitro transcription experiment. The third one is cloning vector construction tools in order to construct expression vector for application in either prokaryotic and eukaryotic systems. But I think to better understand how of what we are able to do at SMALTIS, I think the best is to switch on the second part of my speech and to speak about live bacterial cell analysis. So, live bacterial cell analysis, as you know, is a core research tool for researchers and also for our customers. So, we have set up several preclinical methods to characterize new anti-infective compounds. So, I'm going to present you three of them, requiring, of course, the use of the Tecan reader. So, for those who do not know, to colonize and initiate infections, bacteria must adhere to the host cells. To do this, they use common adhesins such as pili on fimbriae, that you can see on our schematic view. And these pili of fimbriae are hair-like structures that interact with specific host receptors located at the cell surface. For instance, this mechanism is well described for the adhesion of adherent and invasive E. coli strains into the final section of the small intestine of patients suffering of Crohn's disease. But the adhesion phenomenon can be prevented by using compounds able to interact specifically with bacterial adhesins in a competitive manner, and to assess the bacterial inhibition properties of anti-infective compounds, we have carried out specific methods. The first method is an in vitro adhesion assay performed from eukaryotic cell cultures. So briefly, we cultivate epithelial cells to 100% confluence. Of course, depending on the type of infection of disease you want to study, we adapt the cell models. For instance, if I came back to the Crohn's disease, for this disease, for this pathology, we are going to use intestinal cells. So when cells are to 100% confluence, they are infected with selected bacterial strains, with a specific range for 10 to 100 bacterial cells per eukaryotic cells. And after 2 hours of infections, eukaryotic and bacterial cells are stained with DAPI and specific antibody conjugated to lexafluor respectively. And the bacterial adhesion is assessed by fluorescence measurement. So when we carried out this method for the first time 2 years ago, the fluorescence was measuring by using an inverted fluorescent microscope equipped with a specific camera, and now we use the Tecan reader. And I'm going to show you how Tecan reader has improved our efficacy. So as you can see on this picture, this is the results obtained with the microscope. So eukaryotic cells nucleus are colored in blue, and bacteria appear in green. If you look at the picture from the left to the right, you can observe the impact of different compounds on bacterial adhesion. On the positive control on the left, without molecule, when bacteria are cultivated without molecule, you can observe a strong adhesion. But if you look at the 3 other pictures, each of the 3 different compounds tested here have a different effect to prevent bacterial adhesion. So the advantage of the microscope, as you can see, is the visualization of the adhesion phenomenon. But there are a lot of disadvantages. First, picture capture is very long because we must take between 4 to 6 pictures per well in order to have a good overview of each one. Then, once pictures are taken, we must measure by using the microscope software each colored surface areas. So the black one, the blue one, and the green one, in order to make a ratio and to quantify the adhesion phenomenon of each strain, and therefore the impact of each tested molecule. So to give you an idea, before using Tecan reader, we need about 2 days per plate to quantify the adhesion phenomenon of bacteria on eukaryotic cells. But now, if we use the Tecan reader to measure the fluorescence, first of all, the fluorescence is measured automatically. And the device provides us data directly under Excel tabular form, and the acquisition time is reduced to 1 hour per plate. And so if you look at the result corresponding to the previous picture, you obtain directly the percentage of adhesion for each condition. And so, for instance, for the positive control, you can observe that the percentage of adhesion is around 75%, and with the there is a mistake. With the last compound, we have no adhesions, indicating that the compound has the good effect to prevent bacterial adhesions. So, thanks to Tecan for this essay, we have win a lot of time. The only problem is that we have no visualization of the adhesion phenomenon as for the microscope measurement. The second services, the second method I want to talk to you is a method developed to assess the bacterial adhesion inhibition potency of anti-infective compounds. And this method is called an aggregation assay. The objective is to measure how compound is able to interact with adhesins of bacteria. This method is easy to set up because you just need to make bacterial cultures with or without the selective molecule, and to cultivate both under gentle agitation and to visualize the aggregation phenomenon. So as for the in vitro adhesion assay, the visualization can be done either by using the microscope or by using the Tecan reader. So first of all, if you visualize the adhesion with the microscope, on this picture, you can see a result obtained. On the left, when bacteria are cultivated without compound, you can observe the quasi-plane gray image. But if you had a molecule which is capable of interacting specifically with bacterial adhesins, you can observe bacterial aggregation network represented by a less homogeneous gray picture. The unique advantage of using the microscope is the focus scale from 10 to 400. So you can very look specifically on a targeted point into the well. But this is clearly not adapted to this experiment because you must incubate your plate in a separate incubator, and so you must stop the bacterial growth to take the picture, because this aggregation assay is performed during 5 hours continuously. While when you use the Tecan reader and of course the camera included, you can incubate your plate directly into the incubation chamber, and so your pictures are taken automatically. And so you can reduce the experiment time and the cost. And of course, you have plenty of points leading to a better view of the aggregation phenomenon. Moreover, with this device, pictures are always taken at the same point into the wells, and so you have really a better view of the aggregation phenomenon. The only one small problem with the Tecan reader for this method is that you don't have a focus scale, and so sometimes when aggregates are too small, it's difficult to well observe them. To follow, I would like to show you a third, not a method, but a technique we have and the tools we have developed at SMALTIS in order to reproduce and to monitor in real time bacterial infections. To do this at SMALTIS, as I say, we are able to customize the gram-negative genome, and we have constructed gram-negative bioluminescence bacteria that can be used for in vivo imaging, including in deep infection of mice. So, to do this, by using our molecular biology tool, we have constructed our gram-negative luminescence bacteria, and the advantage of this luminescence strains that are these strains can be constructed directly from your own strains. Their luminescence is sufficient to monitor infection in mice, and so to assess the drug efficacy of anti-infective molecules in real time. And finally, because you don't have to sacrifice your animals in order to measure the infection, the bacterial infection, you can reduce the number for your in vivo study. So I'm going to show you how we have developed these tools. So to construct this bioluminescence bacteria, we have developed a specific plasmid containing a very bright photoradius luminescence lux operon from the original plasmid described by Choi et al. In 2005. From this construction, we have cloned different promoters, such as PELAC promoter or PETAC promoters, downstream the lux operons, in order to overexpress constitutively the lux operons. And to select the brightest construction, we have measured the illumination and correlated this luminescence with the bacterial growth culture. So to do this, we have used the Tecan reader in 2 modes simultaneously. The first one was the luminescence mode, so you have the characteristic used for this mode, and the second one is the absorbance mode. By using these 2 properties, we were able to measure simultaneously the luminescence of our construction and also the growth rate of our strains. So, as you can see on this graph, we have obtained 4 constructions which are able to produce different levels of luminescence, and as you can see, there is no correlation with the growth rate. So we have selected the brightest, so here the orange one, and we have introduced it directly into the chromosome of our selective strains. And to do this, we have used a homologous recombination technique leading to the accurate modification of a targeted sequence directly into the bacterial chromosome. So as you can see on the left, we have represented a schematic view of the wild-type chromosome, And thanks to our tools, we were able to introduce specifically between gene A and gene B the lux operon and the most powerful promoter clone downstream the operon. And this leads us to have bioluminescence bacteria, which are usually used in vivo infection model. So I hope that this presentation has given you a good overview of the possibilities that SMALTIS offer to increase your R&D capacity, and especially to perform bacterial cell analysis. I hope too that this presentation gives you a good overview of what the Tecan reader can do to improve your bacterial analysis. And now Christian and me are free to answer your specific questions.
Thank you, Dr. Oberdanner and Dr. Muller, for your informative presentations. It is time for Q&A. If you have a question you'd like to ask Dr. Oberdanner and Dr. Muller, please do so now. Just click on the green Q&A button at the lower left of the presentation window and type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Any questions we do not have time for will be addressed after the presentation. So our first question is, does Tecan also offer dedicated microplates for imaging? And this one is for Dr. Oberdanner.
Oh, yeah, thank you. That's a very good question, actually. And I need to answer it with yes and no. So Tecan does not offer real dedicated imaging plates with cycloolefin bottom or even quartz bottom. But we offer plates that can be perfectly used in conjunction with our imaging module. As it is a cost-efficient imaging module, we offer cell plates, very standard plasma-treated PS plates that perfectly work out for our imaging system and give really a low background in Confluent. So yes, we offer imaging for the Spark imaging module, but not for other imaging systems.
Next one is for you too, Dr. Oberdanner. Do you offer plates with different coatings? No, all of our cell plates are plasma treated, so we do not offer any special surface coatings for various in vitro systems. Just standard cell culture plates, cell culture treated plates. Okay, next for you too. Does the Tecan software for monoclonality verification include an overview of all wells measured at a certain point in time?
Yes. So that question I need to answer with no, because we don't really have a dedicated software solution for monoclonality verification. As I tried to explain, we can offer a semi-automated approach. That means we can perfectly record and archive the images automatically, but the whole analysis process must be done manually. But we save the images in a convenient way, so that allows you to click through all the images very conveniently, but there is no overview about the whole plate per day.
Okay, thank you. Dr. Oberdanner, does the optimal read function also help in 384 well plates?
Again, a very good question, which again I need to answer with no, because the beam diameter of the Spark system is already optimized for 384 well plates, so you can be assured to cover almost the whole area of the 384 well plate. The optimal read function becomes more and more important the lower the well density gets. So in a 6 well plate, I would say it's really a must, but it still is shows a significant improvement also in 96 well plates. So everything between 1 well and 96 well, optimal read really helps to get a better overall result.
And this next one is for you too. Have you also compared signal normalization with Confluent to signal normalization with protein quantification?
Yes, we tried to, or we compared the Confluent normalization to normal protein quantification or normalization done with protein quantification, and we used the BCA assay for this. So we have that data, but not published in an application note. We can provide it certainly to our customers.
We're almost out of time. This will have to be our last question. Dr. Oberdanner, can this system be used in plant research like in tobacco cell lines?
Yes, in principle, that's really a strength of the Spark reader, as the system includes the only air condition available for a multimode reader. We can adjust any target temperature between 18 and 42, and that especially enables the system for non-mammalian cell cultures like plant cells, like various kind of microorganisms, whatever. So that's the clear strength of the system. Yes, you can use it for tobacco plant research as well.
Great. I would like to once again thank Dr. Oberdanner and Dr. Muller for their presentations. Do you have any final comments?
Thanks. No, I'm really happy. I was really happy to talk to you today, and would like to thank you in the name of Tecan.
Well, thank you once again. I would also like to thank our sponsor, Tecan, for making today's educational webcast possible. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 22nd, 2017. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.